Today we are diving into the cell, swimming through the cytoplasm and looking at RNA and proteins and things so that we can better understand and detect and prevent neurodegeneration. We want to keep them neurons happy and alive. Oh, yes, we do. And to help us with that is today's guest from the beaches and laboratories of San Diego, neuroscientist and researcher Norah Al-Azzam. You are a neuroscientist and you work with neurodegeneration, RNA therapeutics. You study proteins, RNA, all kinds of stuff that I just do not know about. Before we get into all the science and all that, first I want to ask, what got you motivated or inspired to even get into neuroscience in the first place?
To even get into science, I think.
Yeah, maybe you were always into it. I don't know.
I wasn't into, so I don't have this on my LinkedIn, but I was actually a political science major in college. So that's why I declared I wanted to become a lawyer
Uh,
my freshman year.
yeah,
College, I went to San Diego State my first 2 years, and then
yeah,
I went to UCSD. But my first 2 years, I was political science. Poli-sci, I wanted to become a lawyer, all that, plan for my LSAT and all that stuff.
yeah.
And I remember I took a class. I think she was Dr. Kelly Duran. She came in because we had to take this class. It's like STEM for non-STEM major. So I don't know if you took
Hmm.
that class, if you had that similar class during your undergrad,
Hmm.
but it was basically a
No.
class to teach STEM to non-STEM majors. So like
Yeah.
communication, the poli-sci, the philosophy, the anthropology, we all had to
Yeah.
take it. And it was a
Uh-huh.
seminar-based class. And then Kelly Duran, she came. We each week we get a new professor, right? So she
Yeah.
came and then she was telling us about her research and what meningitis is and how it's still impacting third world countries and what she does. And like I was really fascinated by the way that she was speaking. And so then after class, I reached out to her and then I was like, I've never been to a research lab before. Can I come? Can I see what it's like?
Yeah.
You know?
Uh-huh.
Kindly invited me to her lab, which then I started to volunteer in. And that's what I started really my science journey. I then, changed majors to biology. I transferred schools, right? I went to UCSD to do a bio degree.
Yeah.
I started to study more sugars. So I started studying these glycan carbohydrates in metabolism and understand their role. And then I started to study them in lysosomal storage disorders. And that's when it got me into neuroscience, was through these rare diseases that impact both the brain function, but also the peripheral function as well. And these are very rare disorders that kids get. And so then I just got into neuroscience then, and then I decided to take a break between going to grad school. So I worked in industry for a couple of, years, I would say. And then I decided to do a PhD in neuroscience. That's how I ended up.
Okay, yeah, you're mentioning these childhood disorders. One of them that I know you were studying was MPS IIIA, Sanfilippo syndrome. Some
Yeah.
people call it the kind of Alzheimer's or dementia for children. I guess it's much more than that. Let's start with that actually, because that seems like that was one of the things that kind of got you started on this journey, and that you were interested in for whatever reason. Talk about some of these childhood neurodegenerative disorders, such as Sanfilippo syndrome. What are they? How do they relate to these adult type dementias? And why are you even interested in it? Yeah.
Yeah, that was my undergrad work, right? So I
Okay.
found this lab, Dr. Philip Gortz. And in this lab, I mean, we work on carbohydrates, glycans, right? These are like
Yeah.
sugars that are not the ones you eat, right? Not the ones that you consume on a day-by-day basis, but they're the ones that are, they're in your cell-to-cell surface. They're responsible for cell-to-cell communication. There's like so many different roles these glycans operate. When it came down to lysosomal storage disorders, they have cachexia. That's what the patients have, right?
Yeah.
And this basically happens is where they have this enzyme. It's missing an enzyme, sulfamidase. This enzyme breaks the sulfate groups from the glycans, right? That come into endocytosis. They go to the lysosome, and the lysosome breaks it up, right? That's normally your
Okay.
function. But in these patients, they don't have this this enzyme. And so all these glycans, they just accumulate in the lysosomes and they cause these storage defects. So that's what they say storage, right? And so they just accumulate and then they cause, damage to the mitochondria. They cause damage, just internal damage within the cell. And these patients exhibit both a proliferal phenotype, so they don't gain weight. They're cacoxic, basically their fat tissues basically burn a lot of fat. They don't store. And from a neurodegenerative stand of view, or from a neurological stand of view, they're basically have almost like Down syndrome. They behave like Down syndrome kids. They're
Yeah.
not intellectually competent. They don't have the communication skills. They don't have the normal developmental skills that you would have, growing up. And so it's both kind of a CNS and a peripheral disease that affects both. And that's
Uh-huh.
what really got me into it. The paper we published on lysosomal storage disorder, that one we did, that one was only peripheral. So we only focused on the peripheral phenotype. We only were interested in basically, this is FYI in a mouse model. Sorry, my cat in the back is like meowing.
It's
I
fine.
just he just wants to get out. That's why.
I
But
thought
yeah,
that was you. I thought you were meowing. I was like,
no,
wow,
I think
I
it's
just
my
met you and you're
cat.
meowing.
He likes to
Whatever.
go out and like this around this time, but he can wait. Oh, he's just gonna keep going.
Fine.
It's
Yeah. No. There were sounds for sure.
yeah, but these patients, they have a peripheral phenotype. So what we did, we modeled them in a mouse model. So we literally made a Sanfilippo mouse model in the lab, created the same enzyme deficiency, and we were characterizing it, and we found an enzyme replacement therapy in this mouse model. And we tried it on the mouse because this was getting like a clinical trial in the patient. We tried it on the mouse, and we were able to rescue the peripheral phenotype, but we weren't able to rescue the cognitive, right? They still showed these cognitive impairments, and that's because the enzyme replacement therapy never crossed the blood-brain barrier. And so that's what got me into neuroscience. I was like, wow, I'm really fascinated on these like drug development aspects, and how can you get molecules across the brain, and how can you actually treat patients in these very rare diseases. Um, but yeah, that gives you kind of a glimpse about it. But the paper is already published, it's out. I can send you the link, you can kind of read about it too.
Yeah. To be honest, I had to toss your papers into AI machines like Gemini to decipher them. I'm like,
Oh,
okay, can you tell me what's going on here? And Gemini's like, oh, I don't know. No, I try my best, but this is where AI kind of helps too. Tossing academic, scientific journals and stuff into those things. I hope it's not de deceiving me. But yeah, it is helping me because these are d deep waters of terminology and vocabulary, that I just haven't really swam in before. I was one of those kids that who's kind of just checked out in high school chemistry, and to satisfy my science requirement in university, I think I took geology or
yeah.
anthropology or something, and I'm finally getting into it now. Um, so yeah, you might need to first maybe teach me a little bit about basic cellular biology, RNA, protein synthesis, the mitochondria, whatever parts of the cell that are relevant to brain health, to these neurodegenerative disorders. This is a huge ass question, right? It's like, give me three textbooks, but yeah, kind of just, if you can get us up to pace, with all that basic kind of
Yeah.
cellular stuff, that'll be quite helpful.
Yeah. So I mean, so yeah, that's what got me into neuroscience first. And then now I do a lot of RNA. So I don't even study these sugars, these carbohydrates, right? So right now I do a lot of RNA work. So from a, DNA, RNA, protein, right? That's the central dogma, I would say. But these RNA in your cell, they're never naked. You can't just see one RNA molecule by itself. They're always attached to there's always a protein decorating it. And that regulates the RNA function, right? That's just how it is, where it goes in the cell, where it's getting synthesized, where it's decaying, where it just goes through these multiple cycles in the cell. But there's these proteins that are attached to it, kind of, like a Christmas tree, right? You have a Christmas tree, and then you put the ornaments on the Christmas tree, and now you have, it's Christmas. That's the same thing. The proteins are these like literal, decoration. And so the RNA is never naked, so it has these proteins. They regulate function. In neurodegenerative diseases, these proteins that's on the RNA, they get mislocalized. they go. Sometimes they're in the nucleus, they become in the cytosol, they move to another compartment, and that causes like aggregation. That causes kind of the whole metabolism of the cell to just like go into overdrive because they're like, what's going on? I have to now clear it, right? So then it signals inside the lysosome, the recycling units of the cell. It signals them in. It comes in and tries to clear these proteins that are getting mislocalized, but it doesn't do a good job, right? It's not, it doesn't do a deep cleaning. They're still there, they're still aggregating, and they're still causing a whole, changes in the metabolism, changes overall in the neuronal toxicity. They become more toxic and more sensitive. So what we were trying to do for my PhD work, I focus now also on like rare diseases. One is Huntington disease.
Mm-hmm.
Right? That's a CAG repeat disorder. We know the cause for Huntington. It's genetic. We know there's repeat expansion. There's adult onset, and there's neurodegenerative onset, and there's juvenile onset. And then ALS is the other
Mm-hmm.
is rare neurodegenerative disease I focus on. And this
Yeah.
one, especially
You love the rare ones.
this one is rare. It's like I think almost like only 30,000 Americans have it. That's like
Yeah.
very super rare in a population,
Mm-hmm.
and probably only 18,000 get diagnosed and prescribed medication. So it's super rare, but majority of it, like 90% of ALS is sporadic. So you don't even know the cause. You don't even know. The patient starts to
Yeah.
show symptoms, and then the progression of their ALS just starts to go. And they only survive like 4 to 5 years, these patients. So this was super rare. We don't really know, but we know the underlining some mechanism which deals with RNA binding proteins. So for this project, and what I focus on is I tried to figure out what causes sporadic ALS. What is like the first event at the
Mm-hmm.
protein level that
Yeah.
causes sporadic ALS to happen? And I wrote a paper on this. I submitted it to Cell, so I'm crossing my finger it goes through. But it's basically I identified these certain proteins, RNA binding proteins, that if you perturb them, if you change them, if you mislocalize them, you start to see early pathogenesis of ALS. And we did this actually in stem cell models. We would model it in a petri dish, I guess, right, where you get stem cells from patients, you reprogram them, you make motor neurons, and then you try to understand it, try to understand the disease, and that's what we did for this project. And I finished it. I was able to find very interesting pathways that if you perturb them, you're going to get almost the sporadic ALS to show up. But this is only in a petri dish, right? So
Mm-hmm.
ultimately, you want to see if this actually happens in patients. But that's kind of the two cases where two RNAs are involved. In the Huntington disease project, we were kind of more interested in about the RNA foci. So these RNA foci, they form, right? These really chunk foci. But again, remember, RNA is not naked. So it's not just the RNA particles, but there's proteins attached to the foci. So the Huntington disease project, we identified these proteins that are interacting with the mutant Huntington RNA. And then now
Yeah.
we haven't wrapped up this project yet, but now we're trying to see if we can maybe remove these proteins by using like small molecules to see if we can actually start to basically clear this mutant RNA in the Huntington. And this we do also in stem cell models as well. So we get them from patients, we grow them out, and we do all of our analyses. So in both of these projects is very much very translational, very at the patient level, right? So, but again, it's all in a petri dish, right? You can't go so far studying something, in these stem cell models. Of course, you want to see if it actually does happen, or you're able to rescue it in the patients themselves, right?
Yeah. I'm not trying to turn this into a science for dummies, neurodegeneration for dummies kind of book, but I want to step back and get a couple basic words that do trip me up, and that therefore might trip up a lot of other people up too. You might be like, "Oh my God, what is this?" Like, I want to talk about the deep stuff, but protein. Protein is this word that the average person just thinks of as coming from food, but it's it's a much broader term that that adds to confusion for people when we're talking about the protein synthesis and these proteins in the body. It's hard not to imagine a nice juicy steak or eggs or something. This is not what's going on. So what the heck? What is protein exactly? And also, what is RNA? I know these are two words that are like, oh, come on, what are you making me explain these things for? But what even is RNA? What even is protein in the general sense? Because if we don't have a grip on these words, a lot of this won't even make sense, probably.
Yeah. Well, if you want to break it down, what I think of, even like when I was studying glycans, which are just carbohydrates, all
Yeah.
of these components are just like building blocks in the cell,
Uh-huh.
right? They're
Yeah.
literally building blocks. So,
Okay.
your DNA, it's a molecule, it makes RNA, you have, translation, transcription, and translation, then
Yeah.
post-translational modifications. So these are like steps, right? You start with
Uh-huh.
DNA, you make your RNA molecules, which then can, ribosomes come in and make the proteins. And then
Okay.
after the proteins are made, then you have these sugars, either phosphorylation, some form of modification on these proteins, right? To give them,
Hmm.
an extra, I would say like extra decoration or like extra something.
Okay.
But
Yeah, the
it's
Christmas
they're
tree.
usually building blocks in the cell. So
Uh-huh.
the cell relies on these, right? It's
Yeah.
every component of them is what makes a cell, right? And what makes some
Every
components of
cell.
every cell is different, right? So in the neurons, they'll have the same proteins. They'll have, right? It's just they're gonna be doing something a bit different. They might be involved
Okay.
moving because remember neurons, they have axons, they have dendrites. Your other cells in your body, which some of them, right, they don't have this component. They don't have an axon,
Yeah.
they have a dendrite. So even if though they might have the same protein, right, different components of the cell, they're just doing different things, right? And the neurons rely on them to do something else. Your kidney cells are doing something else. So it's like, but you need these components. These are we sort of call them building blocks. Like
Okay.
if you want to break it down.
Yeah.
And that's how we really be able to understand these proteins and be able to kind of dive into also their localization. Like a lot of my research focuses on protein local, like my PhD, all focused on like protein mislocalization. Where is it in the cell?
Hmm.
What's being around it? What's in a proximity of it? So it's kind of mapping it to see. And we study this in neurons because they're super fascinating. Um, so that's what I would say.
Yeah. And then in these neurodegenerative disorders, something's going wrong with these proteins or these other parts of the overall factory of the cell, these other parts of the machine or the process. Something's going wrong with this. So seems like what you're saying is something's going wrong with these proteins, and something's going on with the RNA. What exactly are some common problems with the protein? I guess we hear of this misfolding or whatever. We hear about
Yeah,
tau
I'm not
and beta
interested.
amyloid and
Yeah,
TDP-43 and all this stuff. But in the general sense, like what's the common thread between all of this?
for me, I focus on their mislocalization. So for you
Okay.
mentioned TDP-43, right? TDP-43 is an RNA binding protein. It's involved in splicing, right? We know its function. It's if you stain, right, if you want to see where it's localized, it's very nuclear, like in healthy patients that
Yeah.
don't express. Like if you stain, you go to the neuron, you stain it, you see where it's at in the cell, and you see it's very nuclear. But in ALS, it's not nuclear. It's cytoplasmic, and we don't know why.
Wow.
Right? We don't know why. Right? It's not something that's so you can read more about T. That's actually for the ALS project I work on. So we know this protein is nuclear. We know it's involved in splicing. In ALS, it's not. It's cytosolic. Right? It's a cytoplasm. It's aggregating. You see, if you stain patients' spinal cord tissue, so after they pass away, we get spinal cord tissue from them. You section it. You look at their spinal cord neurons and motor neurons. You stain for TDP-43 in the ALS patient, and you see it very cytoplasmic. But in the healthy controls, it's very nuclear. So you're like, what's going on, right? What is going on? And these are sporadic. So that's what I focus on. They don't have a genetic defect. There's no mutation in any of their genes. These patients, they have it, and we don't know if it's due maybe to environmental exposure, Maybe it's due to some form of, aging mechanism that we don't really know. Is it maybe due to maybe an upstream, like something event that happens earlier on that caused this to happen? So that's why I'm interested in these proteins, which are RNA binding proteins, because they get mislocalized and we don't know what's causing them to mislocalize. And so my paper that I submitted, now it's under review, it's literally tackling that question. It's like trying to understand what is an earlier mechanism. What is something that happens before you get TDP43 to mislocalize in the cell?
Yeah. Yeah. You mentioned environmental factors. There's also things like nutrition, toxins, all kinds of stuff beyond genetics that could be causing this neurodegeneration. What are some of your hypotheses for all of this in
Yeah,
terms of
well,
non-genetic risk
genetic
factors?
risk factors. Yeah, actually, that's what I'm going to be my postdoc work. It's literally going to be like that. Because I got interested in my PhD, like I focused a lot on the protein mislocalization, and I would use like different like inhibitors and like small molecules to like show the pathology of this disease to happen in a dish. But what I got really curious about is exactly the environmental and stuff. And those stuff happen actually. So there's this, it's basically neurology and oncology, right? They're always
Hmm.
separate. But you learn a lot from oncology research because oncology is what it's like the mechanisms, the drugs are out there for cancer. It's just a lot compared to what's out there for, neurology, for example.
Yeah.
And so in cancer, so there's this one PI. He's actually my
What's the, what,
sister's PI.
what's a PI?
PI, a principal investigator. Here
Okay.
at
Yeah.
UCSD, he works in the Morris Cancer Center. So Alexander Lud. So my sister's doing her PhD in his lab actually. So she's doing something completely, but I'm learning a lot from her work because she basically is trying to predict before cancer happens, basically. And in order to do that, they do a lot of they call them somatic mutations.
Hmm.
So mutations that happen, they're not at the gametes, they don't happen like genetically, they accumulate through environmental exposure. So her lab publishes a lot on these papers where it's like, oh, what happens to UV light and your skin cells? Do you accumulate these somatic mutations, right? They're called signature mutations that
Yeah.
accumulate, and if they have too many of them, you become and they look at the genes where they accumulate at, and be like, oh, if you have too many of them, then you are a risk factor for developing skin cancer, or you have too many of them, you have a risk factor for developing breast cancer, or something like that. So her lab focuses on that. So what I learned from that, I was like, well, can we apply this concept that we learn in cancer to neurology, to neurodegenerative diseases, and be able to basically screen people before they even develop it based on these somatic mutations, based on these signatures that happen. And so what I think, and what I proposed, to do my postdoc work on, is really to really focus on these mutations that accumulate environmentally, right? It could be anything. Could be maybe somebody that was smoking for all their life and they stop smoking, and then down the line they're getting these symptoms of dementia, or they're getting something, or maybe somebody that got exposed to like they were had almost like their diet completely changed, and then eventually they got older and they're growing all these mutations. Basically, when you're aging, you're growing all these mutations. And is those mutations equivalent to a risk factor of developing a dementia or Alzheimer's? And we don't really know this, right? This is something that's not like known. In oncology, in cancer, they know because they pre-screen the patients that come in. And that's what she's doing. She's doing it from saliva, literally. She's getting saliva and she's sequencing and she's looking, has a computational pipeline built in to find out how many somatic mutations are found. We don't do that in neurology. We don't do that in neuroscience. We don't know why. It's
Yeah.
probably to the healthcare system and the screening, and we don't really do that. Like we don't look for these signatures. But if there's a way where you're able to do the same thing in a population that we call at risk of developing dementia, or at risk to maybe developing ALS, or at risk, you can then get all this information from them, right? But that you have to do that very early, right? When somebody that's like 55 years old or 60 years old, just to see what potential mutations accumulate. Because if they're accumulating these mutations, right, they might be, damaging the DNA, most likely damaging the RNA, and most likely mislocalizing the protein down the line. So it's more like you're looking at it very early on. And that's you can do a lot of somatic mutation profiling. You just have to get like a big cohort of people and do those, analyses down the line. But that's something, because again, in sporadic ALS, we just don't know, right? We see this mislocalization of these proteins, but we don't know if it's like something that's happening earlier on in their
Yeah.
lifetime that's causing this downstream. And there's actually one paper that's published on this so far that did this. It's still, I think, under review too. But I think the field of neurology is going to, neurodegenerative disease especially, is going to switch over. We're going to learn a lot from oncology. Like we're going to learn a lot from cancer. Like, we're just going to learn how these techniques they use, even from a drug point of view. So usually in neurodegenerative diseases, people are like one target. So amyloid beta, right? One
Yeah,
target,
yeah.
one drug, that's it. It's
That's
actually
it.
wrong. That's a wrong like
Hmm.
therapeutic angle. It's not going to be one target. It's going to be like 4 or 5 targets. And then you have to develop what they call cocktail drugs or cocktail inhibitors that target different pathways within the cell to clear a lot of these proteins. Because that's what they do in oncology. They do a lot of this like stuff. They do a lot of these cocktail inhibitors, just to treat a particular cancer type. But they don't necessarily do that in neurodegenerative diseases because we always think it's one target. It's never one target. The cell is going into many changes, and it's not only going to be one protein that you're going to be targeting. It's going to be multiple pathways within the cell.
Currently, what are the ways for detecting these various different neurodegenerative disorders? Are any of them ones that we can just do based off our own sort of subjective experience, some conscious physiological response or whatever, or behavior? I'm guessing it's gonna be more of these things like that you're developing, these biomarker type things where you're getting into this unconscious side of the body. It's something deep in the cell, and this test, just like these cancer pre-screening cancer test stuff, is doing. Yeah. So what's available currently in terms of screening for these disorders?
During my PhD, I wanted to shadow a doctor that treats ALS patients. So I went for like 5 weeks. I shadowed him once a week to see the patient journey. And I was very disappointed like at the end of it. I was like, this is really bad. And that's because the patient come in, they already show symptoms.
It's too late.
ALS, right? They already like, they say, oh my God, my left side of my body, like I can't move it, or oh, I'm having a hard time with my speech. I'm like, that's already symptoms. That's like symptoms. And then they go
And you
in
can't reverse it at that point. It's hard. Much. Is it
already.
even possible to reverse it at that point? Maybe
That's
no, right?
literally what they do. I can tell you that. When I was shadowing the doctor, he was just give them a scale.
Uh-huh.
It's the ALS for each, even in Huntington or in Parkinson's or in Alzheimer's, they all get these subjective scales. They're just questions that the
Right.
clinician asks. Yeah, they're just questions.
Right.
They're like,
It's way
okay,
too late. Yeah.
guys, like, did you wake up? How your sleep? How did you roll out of bed? Can you rate? And then they calculate it. And ALS
Yeah.
have a calculation. If the score meets that after they do the questions, and then they do some. I remember when I was in the clinic, they have them walk around. They do sometimes
Yeah.
a muscle test, like it looks at their muscle
Yeah.
spasms and all this
Like
stuff,
tap their fingers or something. Yeah.
stuff like this. But that's like already too late. That's like the patient
Yeah.
literally coming in and be like, "I'm showing symptoms of some motor defect," right?
Yeah.
That's like too late. So yeah, that's really, it kind of sucks, but that's really the way it is. You get older, you're 55, you're 65, you're 70, you're prone already, like but aging is already, there. And so even some of the patients, like the ALS patients, are like the ones were athletes. They were like they played tennis or they played very competitive, sometimes competitively, and now they're diagnosed with ALS, and you don't know, right? And it's just too late by the time they come into the clinic. and they just get a subjective questionnaire, right? They ask, and then they get diagnosed that day. And then for their treatment response, they do the same thing. So really, it's just screening. It's just really screening. Like the whole system, the healthcare system. So other parts of the world do it like 10 times better, right, than the United States. Um, because, each year you're supposed to get a physical, right? Ideally,
Yeah.
or every
Yeah.
6 months. You should get a physical.
Go to the dentist every 3 months. No, I'm
Right, or
kidding. Yeah.
you should get your teeth
Yeah.
cleaned every 6 months. You should
Yeah.
go see your
Right,
general
right.
doctor, your general physician every
Uh-huh.
once a year or whatever.
Yeah, yeah.
But then even when you do your checkups, your once a year checkups, like they're super lame. Like I do it all the time, the physical checkups, right? They're not. Yeah, they wait you, they do your blood pressure. It's just the clinic is really bad. Like it's just not
Very
like
basic stuff. They give you an eye test, but then if you fail it, they're like, "We'll just give you another chance."
yeah. So it's very like the whole healthcare, and then the only thing they do is then they insurance wise they just bill it to insurance. But that's the
Yeah,
whole.
yeah, yeah. Anyway.
It's not specific. You're not getting a genetic test. Whose insurance company? Not to my knowledge. Even the ALS patients had to pay for the genetic test to see if they have the genetic form of ALS, and they pay for that. It's like
Oh, they pay for that. Hmm.
out of pocket. It's not covered by insurance. So
Okay.
some of this stuff is just like it's horrible, right? Sometimes they find like loopholes where they can help pay them for this like genetic testing, but sometimes it's just the patient just has to pay because it's just not covered.
How would the how would the testing, the genetic testing or the I don't know what you're doing, testing the RNA or something? This is beyond me.
Uh,
Um,
there's companies
how would that differ with neurodegeneration versus how they're doing it with cancer? Are they completely different methods? Are they related or
cancer. This kind is very thorough. Like cancer, they even like if they think it's a like they do like the whole like scans. They do their
yeah.
scans, and if they
Right, right,
find it, they
right.
go in and scoop out the tissue, and they do like a sectioning and all this stuff. In the brain, you
Okay.
can't just go scoop out, a brain. Now, unless if you have like
Yeah.
cancer form, right, that's developing in the brain.
Yeah, you want to stay out
Um,
of there as much as possible. Yeah.
so yeah, it's very like in cancer, and they do a lot of blood work tests, and they have a lot more biomarker panel. They run them. They run for oncology. They run like like panels. In neurology, they don't do that not unless it's requested. That's why we do the ALS testing or even lysosomal storage disorder. Like if you want to know you have a lysosomal storage disorder, or if your child is gonna have it, same thing. You have to get requested. You have to literally like check it off and see if it's covered by insurance. And then there's companies that do that, and they charge like a lot of money. I mean, sequencing costs dropped. So Illumina used to charge a lot for sequencing, and because they don't have the monopoly over the sequencers anymore, you have all these other like little companies popping up, and
Yeah.
they'll run it. They know they do targeted sequencing, so they know the genes, they know where the mutation's happening, and they run the patient samples across it. And there's these
Yeah.
sometimes clinical labs. Like for Huntington, they do a CAG repeat expansion. They have a test for that. So they'll say and they'll tell the patient how many CAG repeats they have. But again, that's kind of too late. That's like when you're coming into the clinic showing symptoms, and then they'll run the test. Um,
Yeah.
so neurology, same thing. You have to do it like once a year when you're doing your checkup with your general doctor. Like you literally either ask them and be like, "Hey, I want a genetic test for this," or "I want a whole genome test." That's the thing. When can we afford whole genome testing for people? It's
Yeah.
when can insurance start to cover it? So that's the thing about this stuff. Is like very much it's not in what they call patient standard of care. Like this stuff that we do in research is very research, right? Like we expect these to do. But if you start to put things in the clinic, then you have to deal with insurance and all this stuff. But think in neurodegenerative disease, you just have to get like you really have to go in and be like, I need ApoE, for example. I need to get checked for that. I need to check for all of, and the patients have to get very like when you get older, right? We're very young right now, so when we get older, you have to get really very educated. I think education is so important. It's like, okay, when do
Yeah.
I have to get a genetic test for this, ApoE, I make sure that I do this and make sure that you have to very like do a whole panel. And it's not something you think about, right? It's more when you
Yeah.
start showing symptoms, and then that's just how it is, really.
Yeah. What can we do to decrease our odds of getting developing some of these neurodegenerative disorders? I know you're mentioning how
Hard
like ALS
question.
is a bit
That's a really
sporadic. It's a bit sporadic, and there are these athletes who are getting in. Even these very fit people get some of this. Given what you've been learning, how what are
it's
some
really
sort of high bet options? Like, if you do this, you're definitely decreasing your odds, or you're likely decreasing your odds, or you're at
I
least
think
gonna postpone it a few more years.
yeah. Well, there wasn't there like a good Netflix show?
There's lots of good Netflix shows.
No, no, but was it?
I
It
know
was
about
like
this.
Red Please or something. It was like people that live past 90 years old.
Okay, yeah, yeah,
You know what I'm talking about?
yeah.
That show.
There's a
I
bunch
said
of
it.
those. Yeah, probably.
Oh, there's a
Blue
newer one.
Zone type stuff. Yeah, yeah,
Oh yeah, Blue Zone. Yep, Blue
yeah,
Zone. They did a whole. There's one here in California too. There's a Blue Zone here. Anyways,
yeah.
they
Loma
look a
Linda.
lot.
Yeah,
Yeah. So they look a lot on like the first thing they look for is of course diet.
yeah,
Number one. And even a doctor look for diet. Like how would you
yeah.
eat diet? Do you eat a balanced diet? And the second thing they look at is of course like exercise and health and all that. And then the third thing is like exposures, which is never
Yeah.
asked.
Right.
I don't know. Like I can walk outside and I get exposed to something and I wouldn't know. And I keep getting
Hmm.
exposed to the same chemical over and over again. How is that gonna, affect me growing up and aging? So it's all about environmental exposure. So it's what exposures do you expose yourself throughout your livelihood. I can give you one case study.
Yeah.
So this is again in oncology. So it's very much cancer, right? Because they do a lot of these exposures.
Well, it's
So
great that you're bridging these two together because a lot of people aren't well versed on both sides of the or these two
uh,
sides of the equation, so that you might come up with some new creative insights, and that's amazing. So
yeah,
yeah, go
so
ahead.
yeah, so women they get their nails done. Okay, I go get my nails done. We expose
Yeah.
ourselves to UV light because we get your nails done for your nails to harden and like the manicures, pedicures done. You expose
Right.
yourself to the UV light that's at the nail office, at the nail salon, right?
Yeah.
And they did this study where they literally looked at women that would get their nails done every week. Like they're like dedicated. Every 2 weeks they're getting their nails done. And they're exposing their hands. They're looking at their hand to the UV light, right? And that UV light is only like 2 minutes, right? 2, 3 minutes. Like maybe sometimes it goes up to maybe depending on what your style is and whatever. And they
Yeah.
the skin cells from these women and they brought them into the lab and they sequenced the skin cells. And when they found
Yeah.
out these women, literally they were looking at risk factors for like cancer, skin cancer. And they found out all this stuff came up, like every single one. And that's just from them getting exposed to UV light in the nail salon.
Yeah.
So that's just one case study. So if you think about it, throughout your day, what are you exposed to?
Yeah.
You're
And it
so
seems like these women who are getting these their nails done every 2 weeks or more, there's a lot more than UV light going on in such a woman. She's probably also wearing more makeup, more of these weird beauty products and shampoos and all
yeah,
this kind
you
of
don't really
stuff.
know, right? Chemicals, right? That's the other thing too. When you get older, you're like, I read everything I buy, the food I consume, where it's coming from, all this stuff. But you can mitigate so much throughout your life. But
Yeah.
it's like even in the lab as a scientist, like I get exposed to so many chemicals, right? Like I don't even know what's going on half of the time, Somebody's doing something and they release a chemical
Yeah.
and I'm getting exposed all the time.
You're gonna wake up one day and look like the Bride of Frankenstein. I'm
Never
kidding.
know, right?
Um,
But you expose yourself to so many exposures, right?
yeah.
And
Yeah. Yeah.
that you don't really account for. And then in the clinic, they don't ask you these questions. They don't
Yeah.
ask you like, do you think you got exposed to something? Like, how long have you gotten exposed to it? What is your even when people that, for example, like smoking, for example, right? You smoke maybe your teenage years and you stopped smoking.
Yeah. Ideally.
That
Yeah.
means you're gonna have a higher risk at the end because when you go in the clinic and they ask you, are you a tobacco smoker? And say no, but they
I
don't
quit
ask you
yesterday.
Yeah, have you smoked, like when you're a teenager, when whatever. Like same thing in a lot of these neurodegenerative diseases, like they don't really ask these environmental exposures. They don't ask questions regards to like they're not very detailed.
Yeah. And now there's all kinds of new stuff coming into the equation. There's various different pharmaceuticals. Some may be helping, some may be harming in some ways. We've got marijuana, now we've got psychedelics. There's all kinds of stuff that we don't really know exactly what it's going to be
Yeah.
doing
And we
to
don't know if
the.
marijuana can cause cancer either. So that's the other thing. No, because
We don't.
that's what
Yeah.
my
Yeah.
sister, her research is literally on that. Literally, it's like tobacco smoking, marijuana smoking, and it's like, do they cause can't we don't know.
Yeah.
So it's like a lot
Right.
of these stuff like you don't really know until you even when tobacco smoking came, like if I asked my grandpa, he was like there were ads that
Yeah.
were literally saying smoking helps. Like your doctor was like, go, like take it, go ahead.
Yeah.
And now you see all these problems. So it's
Right.
like you don't really know. The only thing you do is educate yourself, right?
Yeah.
Is to really have a balanced diet, to practice at least mental health is really kind of I think some components of it. So something I started on the side during my PhDs. I started a startup literally. So now I co-founded a startup called Nanomood, and
Yeah.
it's psychiatry. That's the only thing we
Uh-huh.
focus on. Is we focus on like depression, post-traumatic stress disorder, and we're trying to basically find digital. We call them digital biomarkers.
Right.
A lot of these psychiatric disorders that, to be honest, can lead to a neurodegenerative disorder down the line,
Yeah.
right? So it's all about monitoring.
Yeah. And here you're not checking this like RNA, protein, deep cell
Oh,
stuff. You're checking some other kind of biofeedback. What kind of biofeedback are you using for this?
everything. So like wearable devices, literally, like if you think about it, you're a walking bank of data. Like each one of us is a walking bank of data. Like literally, think about it. And if we wear
Yeah.
a
Yeah.
wearable
We are. Yeah.
device, if we're on our phones, like all of these things are data points. As scientists, we love data points. The more data points you can gather from somebody, the more insights you can get.
Okay.
So when I started Nanomood about like 2.5 years ago, I focused a lot of it on wearable device data. I was interested in heart rate variability. I was interested in sleep cycle and all this stuff that you can gather. Some wearable devices are better than others, of course, that you can gather from somebody. All right. And from those, can you predict their risk factor for insomnia, their risk factor for depression, for post-traumatic stress stroke, all these stuff, right? That you can just get from digital passively,
Yeah.
right? And we're still early on. We're early stage startup, so very early on. The
Yeah.
university backed us up. So right now we're incubating within the university. We're trying to do LLMs, large language models. So we want to do that on. We're like one of the first to do that on like wearable devices because a lot of LLMs are built on text data. So we're doing a lot of those predictive algorithms just to see how accurate we can get. And if we're able to really just get passive data to somebody and track you for all your years till you age, right?
Yeah.
You can get a bit more insight on you as an individual. And on top of it, if you get your like sequencing data done or any of that, you can integrate that to your model and be able to
Yeah.
get more insights. So
Yeah.
to be honest, even though my company is very psychiatry and I love understanding again anything in neuroscience, um, a lot of those insights you get translate over to ultimate other diseases that might occur, but you don't have the data point, and the longer you have the data points that you could tracking these people in. So I would say, like going back to your question, like how do you prevent? How do you do this? How do you live? I would say I don't really know. I think ultimately, if you can get a lot of that data, your own data, the ownership of that, you can
Yeah.
walk into any clinic, right? And they can look at it and they can tell you what you are going to be at a risk factor for, and then
Uh-huh.
how you can reduce it. But we don't have those data points from people. Not your healthcare records are not not all of them are digitalized, FYI. That's the problem. So
Yeah.
again, going back to the healthcare system and all this stuff. But ultimately the future, especially right now with AI and everything, it's gonna change the way we even like treat patients and the way
Hmm.
we even see people and they be able to put them in brackets, right? What's a high risk, low risk, and all that. And that just really you have to be able to not get data right away, like collect it actively, but you have to also collect it passively. And that's just really how it is. Even in the ALS patients, there was one study that got published where they put them all on wearable devices and they were
Yeah.
tracking all these biometrics that came up. So it's like it's becoming a bit more, it will become eventually the norm, basically. That you're just the walking data bank, that you can just lay out your data that can tell you all your history that you had. And that will give you kind of a whole assessment of what you're going to be predisposed of getting and how you can able to mitigate that.
Right. And we don't even know how powerful AI is going to be in 30 years or 40 years, like when you are getting old. And now that you're collecting these data points in your early years, just like, maybe you're collecting tests about your hormones or whatever else, and you have all these different data points. And yeah, it might be useful in some ways now, but there's also this potential of how are we going to be able to analyze this data in 30 years? And you're collecting this when you can. So it's also like spanning it across the time span of your life.
You can understand
Yeah.
aging better too. Yeah, it's just the thing is like the problem is like people don't I see everybody as scientists, right? Every person is a scientist. Because they're part of an experiment or whatever that is. You,
Yeah,
your
yeah,
body,
yeah.
the control of. So
Uh-huh.
I think if people are becoming more aware of their own like data and what they're collecting from themselves, because the healthcare records they're not going to be storing any of that. Like, nobody's going to be storing
Yeah,
that.
yeah, yeah.
It's going to be so much storage in it, right?
Yeah,
But to
yeah.
be able to then store it and be able, I think there's some countries, either Finland or Switzerland, they're doing that where people just come in with their own like they call chips, that has all of their like medical records, their background, what doctor they saw, who they saw,
Yeah,
what.
yeah. I know Estonia has some stuff like that. Yeah.
Yeah, exactly. And then it just because it's the healthcare system is public and it's not like privatized here, so it's very much you walk with your data, and then there's companies like my startup that are there to analyze that data for you, right? If you're doing psychiatry, then we'll come in. But there's going to be a lot more of that kind of revolution into the healthcare system that will start really people that are healthy or we think they're healthy, and be able to like monitor them through years and decades, and be able to get more insight on an individual and be able to really like prevent. A lot of it's going to be prevention. Treatments right now, even though I'm a neuroscientist and I do believe there's always going to be a therapeutic for something, sometimes it's just too late. For some, the disease I work for is very rare. So if you're able to prevent, they're able to live longer. But we're
Yeah.
not there yet. I think I know we're like thinking about, oh, it's gonna happen in this generation. It's not gonna happen in this generation. I think, we're gonna grow older, you and I will be old, we're gonna be living, maybe our grandkids or great
Mhm.
grandkids, they will see the fruit of our labor. They will actually live in that world. But now what we can do is build it for the next generation.
Yeah.
It's not, I know everybody's optimistic and they're like, oh, AI is gonna, no, it's gonna take years.
Yeah. There's a couple more sciencey kind of things I wanted to touch on before we go too. We were talking about how these proteins get misfolded and all busted up or whatever. But this is happening in all of us, even when we're young. But autophagy is one of these processes where our cells and these organelles and these proteins get kind of repackaged and recycled and cleaned up and everything. Um, what role does autophagy play in all of this, and how can we increase autophagy?
Like in neurodegenerative disease. Oh yeah, there's so many
Yeah,
compounds.
yeah, yeah.
Yeah, yeah, yeah. So not only lysosomal storage disorders where I studied autophagy, but yeah, there's a lot of drugs, not a lot. I would say a lot, a
Yeah.
few on the market that are
Uh-huh.
upregulators. And again, this most of them are coming from oncology. Like literally, they're just so everything you see right now. Some drugs you're gonna see that already FDA approved. for one indication, and then they just put them into another indication. So autophagy is one of these drugs that most of the successes came from studying cancer, because cancer cells are smart and they know how to recycle quickly. They have very high
Yeah.
rates of recycling. In
Uh-huh.
neuroscience, it's opposite. It's like neurodegenerative diseases, the autophagy pathways is like takes it time, right? It's like trying to clear all of these proteins.
Okay.
And so they're learning, can we upregulate? Can we increase autophagy, like drive it, the system? And so there's a couple of actually papers I could send you that uses I think they're called nano antibodies, like
Yeah.
small little antibodies that
Uh-huh.
go and target the pocket of a particular
Yeah.
protein and
Okay.
upregulated or downregulated. Um,
Yeah.
and that's one thing, but that's again a lot of it
Yeah.
is used for disorders that need it. For healthy population, like how can you increase your autophagy? That's a good question.
Just normal non-pharmaceutical things. And we hear about fasting and sleep and
Oh yeah, like
coffee and green
keto,
tea.
the
There's
keto
all that kind of
diet.
cheesy
Yeah,
stuff. Is there anything beyond
the
that?
a ketone diet. I mean,
Yeah.
there's already a lot of papers on that. So you
Yeah.
fast
Yeah.
and then you do keto, you go under keto.
Right.
That could also potentially.
Yeah.
But sometimes is you don't want to like overdrive the system. A lot of people do that sometimes. It's actually pretty bad. Um,
Yeah.
in
Yeah.
fact,
Whoa.
damage a lot of your organs in the meantime. So it's not
Yeah,
just
get for example, some people think, oh, more water is better, so they start drinking too much water and just
oh,
flushing out all their sodium and electrolytes, and they just fall
I
apart,
think it's
for
just
example.
a TikTok thing. I think it's young Gen Zs. They're into their Stanley cups and drinking water like every 2 seconds. No, it's not
In my
really
morning routine, I'm drinking 4 Stanley Cups.
drinking water. That no, it's not. I don't really think it's the whole thing. I think fasting has been proven, and
Yeah, fasting does. Yeah.
we learn that not from Western but from Eastern. So like
Uh-huh.
Ramadan is coming. So I'm Muslim, so Ramadan
Okay.
is coming. I think next week. Is it next week? Yeah, it's next week.
Great. I want to do it. I'm not Muslim, but I just want to do it. Can I do it still? Am I
I
allowed?
mean, you can. Yeah, anybody can. It's fine.
But yeah. Talk about fasting a little bit more, and yeah, maybe get into the healthy benefits of Ramadan.
I think fasting that's Eastern because it's
Hmm.
been proven to really help a lot, and especially like we have 1 month where we literally actually no 40 days from
Wow.
sunrise to sunset we don't eat. We don't even eat. We don't even drink water. Nothing. So you don't do any. You don't do none of it until
It's just
it's
like super long intermittent fasting every day for 40
very
days.
long. Yep, for 40
Yeah.
days. And so you are literally, yeah. It's there has to be more research on this for sure. But if you keep doing that, how long does that affect your aging?
How do you feel afterwards? Do people and you and your family and your friends do they tend to act differently afterwards? You can't put it all down
For
to
the
the
first
food.
time,
It might be it's a cultural,
hard.
social thing as well. But
No, it's actually like, and even my grandpa, he does it too. So it's like the older population
yeah,
will do it. I mean,
yeah,
to be honest, I personally like the first 2 weeks is very hard, 100%. Like you're trying to get your
yeah.
body, the
Uh-huh.
same metabolism, the burn rate, the all that stuff. Like you're so used to it right now because you're eating and you're stuff. But when you
Yeah,
completely deplete that, your body's going through a whole like change. Is everything.
yeah,
At first, you have low energy. You have literally low energy. Like there's
yeah.
no energy whatsoever. And then after the third week, your your energy boost is higher. Like
Right.
you're ready. Like you don't even take caffeine or anything. So you're just
Yeah.
like very alert, very like because your body's getting to recycle other things, right? It's
Yeah,
going
yeah. It's finding all
enzymes
these
are
proteins and nutrients hidden away here and there
yeah.
and cleaning everything
So
up and
fasting is
yeah.
proven. There's a lot. There's a lot of research on it. And during Ramadan, I mean, you do see an effect. I mean, some people like lose weight, right? Because your body's
Yeah.
changing. You're waiting
Yeah.
until
Right.
sunset, and right now sunset is like at like 7 or 8 p.m. something like this. So you're waiting a long time.
Yeah.
You're not drinking water. You're not eating. So it's like very much it changes your whole metabolic
Yeah.
in your body. and your consumption rate as well. And when you're ready to eat, because your stomach shrinks when you're fasting, you don't some of us don't even consume as much. It's not that
Yeah.
it expands, your stomach doesn't expand. You actually should shrink it, like it's shrink. And so your consumption is even less. So um
Yeah.
I think long term, I don't know, I have to look into the studies that done it, but long term how that affects aging is definitely like very interesting. Just
Yeah.
and longevity, like does it enhance lifetime? Does it reduce the risk for Alzheimer's? Does it reduce the risk for dementia? And I think we wouldn't know that until we see it, because in the Middle East, where I'm from, right, we
Yeah.
still have dementia, we still have Alzheimer's, and
Yeah.
Muslim people still have like still are predisposed to it. So we don't really know,
Are,
right? I think
are
it's
there any like differences they've seen in the Middle East in terms of longevity or neurodegeneration? It sounds like what you're saying is not a big difference.
yeah, and
But
I think I'm saying it because I'm naive, because I don't think people studied the population. I can tell you what.
yeah,
So, so I go from here to Dubai a lot of times, and this year there's a paper that's going to get published. It's going to be this called the Arab Genome Project. It's basically
interesting. Yeah.
we never sequenced. I never got my. I now got my sequence, but we never sequenced the Arab population. So
Yeah, it's very white thing. It's not just white, but it's an American thing. Yeah.
yeah, it's very much like white Caucasian. And they tried, there's the Latino one, the genome project gonna get updated. It's a reference genome. The reference genome is gonna keep constantly getting updated with different ethnicities and different places throughout the world. But in the Middle East, we haven't sequenced the population. So this year they started to sequence
Yeah.
the population, and they started to find out things that were like completely different, like 100% different. Like because my sister talks about it. She collaborates. She has a project between here and the UAE where they're looking at breast cancer, for example. So here in the United States, women develop breast cancer, yeah, I know at a higher risk, lower risk
Yeah. Yeah.
of developing breast cancer,
Mhm.
because when they have kids and they breastfeed, they become at a lower risk. In the Middle East, it's
Yeah.
the opposite. The women, they don't. Some women don't even have kids. They're like 18 years old, 19 years old, and they're developing breast cancer, like
What?
earlier. And we're like, what is going on? You know?
Are they
So
drinking the oil? What's going on?
probably not. But it's like
Probably
some
not right. Yeah. Am I allowed to say that? I don't know. It's what a weird thing to say.
it's,
Go ahead. Yeah.
yeah, it's just like it's so like environmentally different, right? Their environment, it's like hot. The
hmm
exposures are probably different.
Yeah.
The food as well. So, but right now we're noticing, at least in the Middle East, is like there's different, probably different parts of the world too, right? That we can't necessarily say everybody around the world is going to develop it at the same rate or is going to develop
Yeah.
similarly, because we never studied the population. So going back to fasting and Ramadan, we never studied this population at a longer time to really say that this is actually it. You know, I think
Yeah.
it's really right now. There's a lot of researchers going in and are trying to really discover all these nuances. Um, but we haven't studied the population. So, that's something that I'm very curious to know, because anything you publish in science that's like very novel about a population, like lands in like nature or science or cell, because
Yeah.
you're studying a new population that nobody studied before. So I wouldn't know if fasting and Ramadan can actually like reduce this. You still get the cases, but it hasn't been studied like longitudinally at a larger population to really find
Yeah.
that that is why. But
And it'd be hard to control the variables too, because there are so many cultural differences in the Middle East, and
a lot.
say it's different way of socializing, different way of relating to one another, all kinds of stuff, food, alcohol, drugs. Yeah.
Yeah, and a
So
lot of the drugs are for in the Middle East are narcotics. So even
yeah,
antidepressants,
narcotics.
and you can't take it like in the UAE. If you want
Yeah.
antidepressant, you cannot take it. There's no. It's a
Oh, you're gonna
narcotic.
get locked up in
No,
Abu
it's
Dhabi
literally
jail or something.
only special cases get it. Like here, everybody, like
Okay.
if I go to my general doctor and be like, I think I'm depressed, my general doctor will give me Prozac the first
They'll
time.
give you everything. Yeah.
They'll give you everything. In different parts of the world, they're not gonna. No, a
Yeah.
lot of stuff is very regulated. So that's the other thing too, access to medication, what type of medication you're getting, like so here I think in the States everybody's getting over medicated, like too much. So
Okay.
I think it's definitely different culturally. So you're gonna have different predispositions to these diseases.
Yeah. I did want to touch on the fact that you're in San Diego. It must be a beautiful place to work. Not just in terms of the weather and all of that, but I mean, this is sort of the science epicenter of all this gene stuff and sciencey kind of stuff. You mentioned Illumina, who's headquartered there, along with a bunch of other these science companies. UC San Diego is kind of the premier science university. What's it like doing all this in San Diego, and what are some things that you wouldn't be able to do if you say you were located in the Middle East or you were in, I don't know, Japan or somewhere completely different? Like what's sort of the nice benefits of being in San Diego in terms of all this research?
Yeah, I mean, San Diego, for me, San Diego's second home, right? It's
Okay,
not, of
yeah,
course, not my first home, right? But it's where I came to study. I
yeah,
So it made it a second home for
yeah.
me.
You're like 18. You're like, I want to go to San Diego because it's like one of the best cities. But
Well, I
yeah,
wanted the same. Well, in Dubai, I wanted Dubai is hot. It's very hot. Like
yeah.
6 months out of the year, you should not be outdoors. Like you should be indoors. And FYI, we have vitamin D deficiency, even though we have all the sun. Yeah, it's so weird.
What
Really
a
weird.
weird.
Because don't go out because it's so hot.
Yeah,
Like 6 months out the year, it's
yeah,
130, sometimes 120 degrees, and you don't want to be out.
yeah.
You're indoors most of the
But
time.
very well air conditioned. I
Very
do
air
remember
conditioned.
that.
So I wanted
Almost
to
too
pick
air
up.
conditioned. But go ahead.
I wanted a place that wasn't hot. I wanted to have. And San Diego doesn't have seasons, FYI. It's like
Yeah,
72. Not
that's
bad.
perfect.
That's sure it's
Yeah,
like perfect. Sometimes it gets to low 60s, and that's like crazy. And sometimes it rains, but the environment is nice. It's very, they call it chill. I don't know if that's something that's
yeah.
not the right word.
No, yeah, I know. Chill. I'm from California,
But it's
so
like very chill. Even the scientists are chill. It's very chill. So you can walk into a lab and literally, like, one scientist just literally went surfing. They put their board in the lab and they walk in and with their flip flops and
yeah.
sand in the lab floor. And then you're like, is this like PPE safe? Um, but it's like very chill. So super
Yeah,
like, I like it because it's not like cutthroat. Like I
yeah,
could have went, right, I had another option. I could have went to New York, NYU, and did my PhD
yeah, yeah.
on
Because the East Coast, like Boston and New York, are these other kind of hubs for all of this,
favorite.
right?
They're
For the
still
opposite
fun.
weather, the opposite kind of vibe,
Yeah,
probably.
still good, right? It just
Yeah,
depends.
yeah,
I wanted not a cutthroat vibe. I wanted to still be competitive in the research. I wanted to still publish. I still wanted to like have career opportunities, right? If I
yeah.
don't choose academia, maybe I want to go to industry. And I started my own company, so I was like, this is great. So like I wanted the flexibility to like pave my own path. So I think very much San Diego it doesn't give you the same. So if I went to SF, right?
Hmm.
It's the same
Yeah.
same thing. But SF, you get a lot of opportunities, 100%. Like there's a lot more
Right, right.
companies, there's a lot more. You can raise venture capital really well in SF. You
Uh-huh.
don't raise venture capital in San Diego. There's no way. There's not a lot of, of that. So,
You gotta go down to Tijuana to get all the venture capital, right?
really. No, you have to go to SF
I
or you
know.
have to go to Boston
Yeah, yeah.
and put your ideas and stuff. But we still get like attention, right?
Uh-huh.
And partly
Yeah.
we recruited really good professors. Like they're very selective. They picked like, Rob Knight, who's like really great in microgut biome. Like he's world renowned.
Yeah.
Um, Wang with wearable sensing, my PI, Genial, like very good at recruiting these really good, they're very big names, right? But do research in a way that's like settle. Does that make sense? So it's not like not very much how should I say, what's that word for it? Like not flashy, but not like too much in your face.
Yeah.
Because sometimes it's like that, right? You have these
Yeah, it's
MIT
chill. It's
or
chill, but
Harvard.
it's got something good going
And it's
on. It's just
good.
it's the right balance. Yeah.
It's the right amount. Yeah, we still get newscasts and we still get people coming at us and asking us questions. So it's not like we don't get the publicity we need. We do get it, but we're not very much uh like cocky about it. We're not very much like, oh my God, like
Yeah.
very hot shot about it. Very subtle. So I really like the environment. So that's why I wanted to do my research here. And then the companies I worked for, right? I worked for Abbott, Inhibrx, Neurocrine. So I worked for the companies, right? A lot
Yeah.
of the people that were in these companies either were associated with UCSD, so they had the same vibe, or were recruited into these companies. And I worked for So when I first started, my first company I worked for was Inhibrx, and it was like the lab I worked for, Kelly Duran. The people that were in that lab started to work at that startup. So it was like we all knew each other. We all got along well with each
Yeah.
other. So even the biotech community, right, in the
Mm-hmm.
pharmaceutical community within San Diego, everybody was like family and everybody's like friends of friends.
Yeah.
So we just kind of like all vibed together. And I think and that's just we still produce like good research. We still,
Yeah.
commercialized products, right? We still worked, but we do it in kind of in our own way that's super unique to San Diego that I don't think any other I mean, I collaborate with so many PIs, right, throughout the nation that I don't think any other place throughout the nation does it the same way we do it.
Yeah, because there are all these great places in the world to be in. How lucky to be in a great place that actually has great job opportunities and people and all of that as well.
It's
I
really
got
difficult.
ambulances over where I am. They're just never stopping.
Where are you at? Are you in
Medical.
Japan?
Yeah, I'm in Japan. I'm in Osaka,
Oh my God, I
Japan.
went to Osaka like 2 years ago.
Really?
I loved it. Then
Yeah.
went to Kyoto and then went to
Yeah. Yeah.
Nara. So we went to
Yeah.
all these places. I want to
Yeah,
go
speaking of
again.
chill, they're much more chill here than they are in Tokyo. I like Tokyo, but it's not Okinawa, but it's something in the middle, I guess, between Okinawa and Tokyo. Yeah.
I love Japan. My brother is an anime artist.
Yeah, wow.
I don't know. So so he wants to go again because he wants to publish his work.
Yeah.
But and I heard the yen dropped. So
Yeah, good for you, but terrible for me.
so I heard
Like, why am I even working? What am I even doing here? Getting paid minimum wage.
I heard it's good to go visit right now because
It's
lots of
really good. It's really great from your perspective. From our perspective, is like we are trapped in this country because this money is becoming worthless.
oh no,
Um,
I'm so sorry.
no, but for you, but this is definitely the time to come to Japan because the dollar is freaking. I know people are complaining the dollar's not worth anything. Just take that money out of the U.S. and you will feel the power of it.
Oh,
Yeah, yeah. Around the it's strong. We're getting into turning into a financial podcast now.
I do
Yeah, I'm from Bakersfield, and then I went to in university. I was in the Bay Area. So yeah, and I'm in Japan. San Diego's great though. Um, and then your sister, I guess, kind of followed you there. You followed her there. I guess you're talking about family being there, and now literally family is.
yeah, my parents. So when I moved to America, my parents were like, I want to go there too. So like, eventually,
Oh, you started it, and then they
yeah, I
came.
went. I did my undergrad, and then my sister at that time was like, yeah, I should come back and like do my PhD. Well, she worked. She came. She found an industry job, and then she was like, I want to do my PhD now. She worked at Eli Lilly, and then she came back um to do a PhD. I was like, oh great, no worries. But my parents decided to come because they want to be close to me because I've been for so long, right? So
Yeah.
they bought a home, so they come visit like every couple of months. They stay for like 2 3 months, their visa, and then they go back. And I
Oh,
go visit them too. They can't stay, right? My parents are retirees too, so they're pretty old. So
yeah, yeah.
they do come visit me, and my brother also came. So it's not bad. It just kind of gets you don't want to. I'm very family oriented, so I do
Yeah.
like to have my parents around, and they're very motivating. I don't come from a family of scientists. I think my sister and I are the only
You
scientists
started a new trend.
in the
Yeah.
family. Yeah, so we started, and all my family there in the Middle East, they're like real estate and they're oil, as you
Yeah.
said, and they're not, there's my uncles,
Yeah.
right? They're not,
Yeah.
but both of my mom and dad were teachers. So my
Hmm.
upbringing was a bit different. So maybe that's why I turned out to be like this.
Yeah.
Um,
Yeah.
my cousins and my uncles, they're very much like business and real estate. like all this stuff that to me is great, but for them,
It's great for them
but
too. They're lucky because now they're getting into their older years and they have a
they have something.
neurodegeneration expert to guide them in their older years, hopefully staving off some of that dementia and whatnot. But there's only so much we can do, I guess.
Yeah, so yeah, so I definitely it's a bit it was nice to have a community, what I'm trying to say.
Yeah,
And
yeah.
to have a community outside the family too, with a bunch of
Right.
scientists that are coming in.
Yeah.
And it's been nice. Like I haven't had everybody's welcoming. And I think San Diego is it's gonna become a melting pot with a bunch of cultures, like everybody from all over the world. That's how you do science, really. It's not like driven by one country, one culture. It's gonna be a multiple. Everybody's coming in so
Yeah.
many different diversity and that's so important, right? So that's why I really like San Diego and I like the diversity we bring because other places it would not have been like that at all.
Yeah. What's kind of currently exciting you most about all of this? I know you're talking about these kind of tests, these biomarker tests or whatever for various neurodegenerative disorders. That's something you mentioned, but yeah, what's kind of like, oh, I'm really excited about this. It probably connects to this paper you just submitted to Cell,
Oh
but
yeah.
yeah.
Um, I think for me in neuroscience, what I'm very excited about is I definitely want to translate my work to the clinic. That's like number one. Like if there is a way where I can translate some of this work
So
that's
then it
not
can like actually get you can see it
make an
directly helping the
impact,
people. Is that what that's
it
about?
directly can help. Because if it's just gonna stay in, a paper in Cell or a
Yeah.
paper in like it's
Half
not
that
gonna do
gets
it.
lost, the internet's not even archiving it. It's just like just
Oh,
hidden away in some storage shed in, you know,
and then you get a news release, maybe like when my paper gets published, and then somebody reads it, and oh, we should definitely do a news release on this and whatever.
yeah.
But like, it's not gonna make an impact. Is what I'm trying to say. So basically, I was able to discover an earlier pathway, a protein that gets to be. It's called this protein is called SMN.
Yeah.
Um, that if you in some sporadic cases, this protein is downregulated, so it's lower expressed and then TDP43 gets mislocalized. That's what I was able to show. So when
Okay.
I decrease the levels of this protein, TDP43 becomes cytoplasmic. When I increase the levels of this protein, TDP starts coming in the nucleus. Now, think about it from a screening point of view. You could screen for this protein. It's not hard. It's not. You get a blood work test and you run the assay for it, and then you could
Yeah.
in this patient if this protein is downregulated or upregulated, and if it is, treat it with a drug that could elevate its levels.
Okay.
So, but will that translate in the clinic? No, that's
Yeah.
gonna take years.
So it's like you're screening for this, and then you're also hoping to or trying to, or maybe even already doing, um, introducing new things into the system. I don't know if it's kind of something about RNA editing, or you're just bringing some drug in, or what. But that's kind of changing the status.
Yeah. So for the last ending for my paper, which I'm really excited about, so you know SMA, spinal muscular atrophy.
That I know the term. I don't know anything
Okay,
about
spinal
it, but
muscular
yeah,
atrophy is a developmental disorder. It's literally the kids, they get screened for it when they're born if they see something,
yeah.
babies, and then they treat it with an ASO,
What's an
and
ASO?
anti-nuclear sense,
Okay.
something like this.
Yeah.
So this is
Everyone listening to this episode is probably gonna be like, "Oh my God, this is like a explosion of science words in my head." I guess it motivates us all to like start, getting into all this stuff because I feel way behind. But yeah, continue.
yeah, so it's a developmental disorder, spinal muscular atrophy. It's where the SMN levels are lower. And so they know that because of the gene defect. They can see the patient, the babies that are born, they
Okay.
have the gene defect. So if they have it, they treat it. That elevates the level for it. And then the patients, the babies never develop spinal muscular atrophy or they get slowed down in progression or they might not get it. But this has been approved probably like recently, like probably like 4 years ago, I think. That's the
Yeah.
drug got approved. It's the ASO for SMA. I think it got approved like 20.
Searching Google.
Let me see. When did it get approved?
20 something.
either 2019 or
Yeah.
2016. I have to check. But
But in the
yeah,
past
it's been approved.
7 or 8 years.
Yeah.
Yeah.
So I can send you this paper, Cure
Okay.
for SMA. So this is
Yeah,
the
yeah.
paper that got released. It's Cure for SMA. Everybody got it's like just the release for it. Um,
Okay. Yeah.
so you can read about it. And so this one, these individual SMA, they have at least one or often copies of a single gene called survival motor neuron gene 2. And so there is already the first FDA approved therapy for SMA is an antisense ASO approved for all ages and types of SMA. So this is actually
Hmm.
groundbreaking. When this came out, everybody was like super, excited about.
Yeah. What's the most exciting thing about it? I'm not tuning out, but I'm a little bit overloaded by some of this terminology. What is like kind of what is the big breakthrough thing about it?
So there's a cure right now for SMA.
Uh-huh.
Right?
Okay. Okay. Yeah.
So it's December 2016. That's when it was.
Hmm. Yeah.
And now you can treat if they have the gene defect, they get treated
Yeah.
with it, and then it's fine.
This is a childhood kind of
Child,
neurodegenerative disorder.
but
Okay. Is it
right,
fatal? Do they usually die from it?
I
Or
think after
because a
uh,
lot of these childhood ones they're just gone by the time they're a teenager and
probably
stuff.
teenagers. Yeah, 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 I think you're right.
Yeah.
It just
It's
came
like one of
out.
those.
Yeah,
Yeah.
it's one of those. And so that was really exciting. It's basically
Yeah.
SMS
Definitely.
level
So it's a kind of a cure, a potential cure for
cure.
this.
So I was like,
Okay.
well, in my sporadic patient lines
Yeah.
I saw SMN level reduction. Like it was lower. So
Yeah.
that's what got me curious. I was like, what's going on? Right. And so then I was like, okay, well, if SMN levels is lower and FYI, if you read more, there is a similarity between SMA and ALS. They share
Wow.
similar molecular pathways. They have almost, there's at least so many papers I can send you. They have intersecting.
Yeah.
One is developmental,
Uh-huh.
or is neurodegenerative, but there's intersecting pathways.
Right.
And one of those was the reduction of SMN levels. And so
And
then
one
I
tends
was
to
like,
happen in older age and one tends to happen
that
in
is,
childhood.
yeah.
But by learning about this, and if we can get a cure for this, that kind of gives hope for maybe we can start to figure out what's going on with this ALS thing, and then we figure out the
That's what
ALS thing, maybe we can start figuring out this other types of dementia things.
well, you learn, right? You learn from one disease and move to
Yeah.
the other one. That's what I did. So my paper really was exciting because it's like that's literally the last ending of the paper was like we don't know if the approved SMA drug ASO can work for ALS. And right now I'm
Hmm.
doing the experiments in case the reviewer asks for it because that's what they usually do to see if we're able to rescue right using this approved FDA approved drug for SMA, and can we use that in ALS? And can we
Yeah.
rescue it in the sporadics at least? And I think that's just that's the most exciting thing I'm looking for. But of course, I won't able to see that in the clinic, right? It's
Yeah.
like if I was able to find something very cool and exciting, then somebody does, a preclinical study and then a clinical study. That's gonna take like 10 years. And then they put it in the patient. Um, or maybe 5 years. I don't know. But that's
Yeah.
what's exciting about research. Sometimes you find something that's really exciting that you can link, two different diseases, find some commonality, find an approved FDA drug for one, and see if you can treat the other. Um,
Yeah.
and that's what I'm excited about. But of course, I won't be able to see this in the clinic. So the patients, the ALS patient,
Yeah.
they might not get this because you have to do a whole clinical FDA approval for this. But I think
Yeah.
sometimes, some of these things are really exciting when you find them as a scientist and you're super excited, but
Yeah.
it's always that roadblock, right? It's like, is it
Yeah.
really going to make the impact that you want to see? And that's partly because, you have to go through regulatory and FDA and all that stuff.
Yeah.
But
And it's exciting to be doing the research, but it's also like kind of frustrating to be trapped behind the research door. It's almost like you're a chef and you don't get to see the people eating your food. You don't even get to talk to the waiters. You're just in there doing your little small piece of the thing, but you want to go throughout the entire house and explore. Oh, how is this affecting everybody? I want to play a bigger role in this, but there's only so much you can do, I guess, for legal reasons, but also just for you're only one person.
Exactly.
Uh,
You're only one person. Yeah. But I think that's really exciting. So, that's the thing I really wanted to see is like if you're able, and I think a lot of my past research, I have always looked into connecting disorders together, right? And learning
yeah,
from one neurodevelopmental to neurodegenerative, and there's common, molecular mechanism. And if there's one that has a drug for it, can you use it for the other one?
yeah,
Is that a potential therapeutic? And I think sometimes it's not. Sometimes the mechanisms might be different. Sometimes you have to, as we talked about previously, you have to think of different modalities, like different pathways that you have to upregulate
yeah,
within the cell because neurodegeneration, so it's one of the hardest, I think. I think neurodevelopmental, there's always more FDA approved drugs. There's always something going on. But when you get older, it's a lot more, you have to think about a lot more mechanism that
yeah,
more than when a neurodevelopmental disorder it's completely
yeah.
different,
And,
right?
and this might be totally out of your territory, but what can these RNA editing therapies like CRISPR do for neurodegenerative disorders? Do you know anything about that? Or is there anything going on with
Oh,
that?
is it because the sickle cell CRISPR therapy got approved, right?
Yeah,
Yeah, everyone's super excited about that, right? It's groundbreaking for sickle cell. Um, I think it's would be interesting to able to
yeah,
use CRISPR, right? We use it all the time in the lab for everything.
yeah.
To
Really?
model
Yeah,
diseases, but in the clinic, as I said, we don't know the effect of CRISPR necessarily and the off-targeting effect because it has off-targeting sometimes, right? It's not a
yeah.
clean system to use.
Uh,
And also the delivery mechanism for it, especially reaching deeper like brain parts in the brain, like the striatum, like all of those like parts of the brain. You have to think about delivery.
yeah.
Like, how are we going to deliver it? Is it going to be like an injection, right? They have to do a whole
Right,
surgery
right, right.
stuff.
Yeah,
It's not like a pill that you take and it's like, oh, okay, I'm done. But it's more about
yeah.
the delivery, the off-targeting effects that could happen. But there's a lot of potential in it. There's a lot of potential in it with AEVs, those ones like the delivery mechanism for those. But there's definitely there is interest. There's 100% interest for a lot of these diseases that you can potentially use CRISPR to recruit different RNAs to knock down different RNA targets that you don't want. So there's definitely a potential for it. I just think that the delivery is going to be
What
important.
is a normal? I'm completely like I haven't really dug into all this RNA editing stuff or whatever, but what's the normal delivery mechanism for this outside of the brain?
So for sickle cell, right, because it just got approved, this one is like, I mean, the first gene therapy to treat patients. This is like super groundbreaking. The
Yeah.
delivery mechanism for these, at least for this one, let's see, what did they use? On blood stem cells. I think they just did a whole injection. I think this is an IV
It's an
injection.
IV injection. Yeah.
Yeah, for these
You
guys.
can't do that in the brain now. Even if you could, like how do you
It's
locate in
not.
the right exact
No, this
spot?
one you have to. So this one is the first approved CRISPR treatment for sickle cell disease. It's like super world renowned. It's November 21st, 2023.
Whoa.
That's when they released I put it in the chat. That's when they
Yeah.
released at least Nature released the thing for it.
What are
Yeah.
some of the before we close this off, what are some of the big misconceptions or myths about these neurodegenerative disorders that you think, okay, stop thinking this, guys. This is not correct. Is there anything like that that you want to share?
So misconceptions. Oh, there's this one about like taking B12.
Oh yeah, yeah, I've heard that one.
Okay, no. This one, no.
No, don't do
Definitely
it.
not. There's B12. I mean, there's a whole paper about this, I think. So people are trying to overdose on B12 or you can't even overdose on it, but
Yeah, you're just
it's
gonna pee it
like
out with your money, but yeah,
yeah, it's like it's stuff like this, like yeah, all these supplements that you think that potentially might, how should I say, like increase, your synapses, or having like higher pruning, or like whatever it is that you think
Yeah.
actually, or
Right.
it enhances your cognitive
Yeah. There's like
performance.
no regulation in that industry anyway, so people just say
That
whatever they
industry, there's
want.
no, so I that stuff I wouldn't, that like call like
Yeah.
misconception about taking B12, and it should like, help you cognitively,
Yeah.
enhance, and like all this like, or get more energy, or just, to be honest, I I could go in the lab right now, take B12, and I would put it in my cells, my culture, and I would see how they react.
Yeah.
I'll let you know.
Are there some supplements though, or nutrients that do that you think might be useful? I would imagine things like omega 3s, and there's some basic stuff that everyone throws out there that like couldn't
Maybe
hurt, but yeah, is there
hurt,
anything?
right? And these things don't hurt, right?
Yeah.
That's where you get your blood work done once a
But it's
year.
not going to save you. It's not going to prevent.
It's not gonna,
Right.
as
Yeah.
long as you're not deficient in some of that. Some people are deficient in iron,
Yeah.
all that stuff.
Right.
But
Magnesium, some
they,
basic
yeah,
stuff. Yeah.
you would know this with your blood work test, right? And so like
Right. But you don't need to
the
go
doctor
go
will
throw in a we're talking about Stanley Cups earlier. Drink a Stanley Cup of
drink a lot of
uh
water from your Stanley cups.
or
Yeah,
a B12.
yeah, stuff like this. I wouldn't necessarily. I would take with a grain of salt. That's what I would say. I wouldn't
Yeah.
necessarily say it's gonna cure. I wouldn't necessarily say it's gonna prevent. I wouldn't necessarily say, a lot of these things. One, they're not really thoroughly researched. Two, they
Yeah.
haven't been researched at a longer time with the same
Right.
population. Like a lot of these longevity like studies, like they go maybe for like 3, 4 years, and that's it. They're not studying populations longer than that, because you need to
Yeah.
get the paper out and publish it.
Right.
But
Right.
to be honest, and sometimes it's all like bad science miscommunication. Like I think we, as a scientist, we do a horrible job at like communicating our science, because it's very complex, like how you said, super complex, super complicated, and you're trying to communicate with the public is super hard
Yeah.
because we're super like caught up in our terminologies and stuff.
Yeah.
So a lot
Yeah.
of it is definitely the miscommunication aspect of it. And so everything that comes out from a research paper, people get super excited, and they're like, I want to do this, I want to try
Yeah.
this, and it doesn't work. Or like it doesn't, or
Yeah.
maybe it's not defeats not the purpose that the paper was trying to cover. So stuff like that, I like the
Yeah.
misconceptions of taking vitamins. Or so, of course, you want to have a healthy diet, right? That's super important.
Yeah.
Sometimes
Yeah.
vitamins, when you take them, they don't even get digested. So
Right.
that's
Right.
the other thing. So you're just taking them,
Yeah.
and they're not even being absorbed in your body. So what's the point? So
Yeah.
those are some things.
But there's also kind of some um, distrust or fear of pharmaceuticals too. Not with everybody, but it's a huge part of the population. I might be included. I'm always like a little bit skeptical of, is this pharmaceutical really doing what it's saying it's gonna do? Is it gonna secretly harm me in this way or that? What can people do to gain a bit of confidence in these therapeutics that you're helping develop?
So side effects profile. There's always going to be side effects, right?
Yeah, right.
Any drug, like, but some side effects are not bad. Like if they need it, like the patient need it, like, like side effects.
Side effect, you're rich and handsome. Oh, great.
No, it's more no, it's more like, the patient constipation, or maybe they have sleep disturbances a little bit, or maybe they gain weight,
Yeah,
or maybe they,
right, right.
like, stuff like this, like, yeah, that's expected. Some of this. But things like high risk for thyroid cancer, or like stuff like that, then I would be worried about it. Like I would not take it, but it's like
No, no,
it
no good.
depends. But sometimes if it's like diseases that like very rare, and this is the
Hmm.
only FDA
It's the
approved,
only way.
it's the
Yeah,
only way, then sometimes the patient just have to, there's that trade-off. Um, but I think it's really a lot of the patients have to do, the people have to read, get educated, understand the terminologies. There's just so much, reading that you can do to really learn and educate
yeah,
yourself and ask these questions, and make sure you get from good, reliable sources.
yeah.
Um, and also, um, people go to AI and they put a couple of things in there. AI,
Hmm.
I would say, in terms of
Yeah,
accuracy, depending
right, right, right.
on what you're, because sometimes I look up stuff, when I use AI, and I'm just like, wait, did they describe this disease wrong? Like, this is not how
Yeah.
to describe it. But there's nobody going in there like proofreading all this stuff. That's the other thing too about we're doing in the scientific community is we're trying to find like researchers that used AI to write their papers. You can tell. You can tell
You
if
tell.
you can
You
tell.
get email. I get so much AI stuff. I'm in Japan, right? And I'll get these emails from these Japanese people. I'm like, your English suddenly got really good. Hmm.
Oh yeah, it's getting really good. I mean, also like AI for scientific figures.
Yeah.
Actually, there's a whole like paper that just
Yeah.
got retracted just because of that, because the AI just did not portray the scientific photograph correctly. So I think also be worried about these things.
You started off on this journey in through neuroscience with sugars and carbohydrates and stuff like that, glucose. Has that come full circle at all, where you're starting to explore that again?
Studying glycans.
Yeah, that one.
Yeah, carbohydrates.
No, it's your old lost baby. It's never coming back.
Yeah, because I studied these for like how we started off, like studying it in lysosomal storage disorders. But now it's not. But there has been a new field that started
Yeah,
like recently, um, where it's RNA and glycan.
yeah.
There's only one. I think one paper that got published. It said it broke the central dogma. So it basically is saying that glycan, carbohydrate, these like sugars, can decorate RNAs, which
Yeah.
everybody is like, really? What? That doesn't make sense because glycans come later in the in the central dogma. They're after the
What is a glycan? I know you're like we got to stop this thing eventually. But what is a glycan?
glycans are just sugars. They're like
Okay.
they're monosaccharides. They're like I said, glucose, fructose.
Oh, these are all
Right.
glycans.
These are carbohydrates. They're sometimes come in one chain, two sugars attached to each other, or it's a long chain where it's like so many sugars attached to each other.
Yeah.
So then, yeah, so it's like, but they usually come after the protein is made. So once the
Oh,
protein
okay.
is made, the glycans come in.
The cells also making this stuff?
Yeah, glycans are made. Yeah, there's genes that
Okay.
make these glycans that happen. They decorate the cell. They usually decorate the cell matrix, so outside the cell. They're kind
Okay.
of these decorative, and that's how we learned about COVID, really. So when
Yeah.
COVID came out, everyone was like, "Oh, these spike proteins." That's what they called them. Um,
Yeah. Spike proteins.
yeah, everyone was excited, but it's just that because the virus is so smart. The virus, what it does, it decorates itself with
Yeah.
carbohydrate. So it's kind of like a Trojan horse effect. So when it comes in the cell is like, oh, this is a part of me. So I'm gonna let the membrane open up to let the virus in. But it tricks the system to thinking it's a part of it, like a Trojan horse effect. But these glycans are very essential. They're really great. And they're responsible for so many things in the cell besides immunology and all of that. Is what we started to know them as because with COVID. But when it comes to RNA, like coming full circle, there's only has been one paper published that basically said actually RNA it's called what they call it sweet RNA. They're saying that these carbohydrates are actually decorating the RNAs. They're actually modifying these,
Yeah.
which is something in biology we're like, what? That's not how we think about it. There's only one paper released on it.
Yeah.
My PI at that time, he's like, "Do you want to do a project on this?" Because I have a very strong, background,
Yeah.
and now I'm learning RNA background. And he was like, "Do you want to do a project on this? And you can pick a disease." And then I was like, "I'm good, thank you," because
I think you secretly you just want to be a decorator. You want to decorate Christmas trees and cakes and things. You use the word decorate a lot, so I'm thinking, are you a decorator
I think it's
behind the
how you
scenes?
explain it. It's harder to
Yeah,
explain.
it is. It
I'm trying
is.
to find like terminology to explain
That's
it.
great.
I think it's easier to explain because I'd be like, wait,
Yeah.
how does that make sense? But yeah, I
Yeah.
mean, for me, it's like I would love to study like glycoRNA. It's a whole new field right now.
Yeah.
Like a couple of professors that are researching this topic, but
Yeah.
it's a new field. It's very new, And sometimes it's risky as a scientist to go into a new field.
If you could like suddenly
Um,
just download a packet of an entire field into your brain and you can use this with your current knowledge, like what field or what kind of thing would you want to just be able to boom, just download?
Field I want to go into. It's hard.
You don't have time to get into it, but you're like, oh, if I had this knowledge, ooh, I could do some magical things.
Yeah, I mean, for me, again, I was not a neuroscientist. Like I was a glycoscientist, and then I switched into neuroscience. So I had to learn a lot of neuroscience terminology. And I don't think
Yeah,
I'm very molecular. So the thing about me is I'm a very molecular neuroscientist.
I
So I
know. I
study
noticed.
more than molecular. I'm not one of those neuroscientists that are like, oh, let's talk about behavior and behavioral testing and like so
Yeah.
sometimes when I see these stuff, I'm like over my head. So
Oh, yeah. It's the
yeah,
opposite for them. Yeah, they're
the
like, you're over
opposite.
my head. I'm over your head. We both are
Yeah.
over each other's heads.
So for me,
Yeah.
I would love to like if I were to read out like I'm trying to touch base with like because I want to communicate to other neuroscientists because I'm super molecular and the people in my PhD cohort they're very behavioral so
Yeah.
they like to do with they work in vivo too they work with a mouse model they work with a rat or they work with like monkeys and stuff so they're very much like hardcore what I call it hardcore neuroscience
Yeah.
I would love to like be able to communicate with them I think
Okay.
and learn
Yeah.
the terminology a lot more because
Yeah.
what I study is very molecular, but can you
Yeah.
connect what's going on molecularly to behaviorally? I think would be great.
Yeah.
But I would love to kind of re-download that into my head so I can be able to communicate. Even scientists have a hard time communicating between each other. That's always on.
Yeah.
So I would love to kind of learn that.
Yeah. Well, thank you so much for teaching me some of this hardcore science. A lot of it was over my head. But I guess I'll re-listen and try to figure some of this out. It does motivate me to pursue this path a little bit more, and it does give me more faith in these RNA-based therapeutics, this weird cellular stuff. I tend to focus on the behaviors, maybe nutrition, kind of everyday stuff. But you're showing us these other tools like these pharmaceuticals are weird little I don't even know what they are exactly. Some things we put into our veins or something to help with things like dementia. So yeah, thank you for that. I probably mischaracterized what you're making there, but yeah, it's not just exercise or sleep or something, which of course are very beneficial, but we also have these other therapeutics that are being developed in labs that could help. So yeah, thanks for researching this and thanks for trying to teach to me. I think I got as much as I could get from it. I should have not been sleeping through that chemistry class back in high
No
school.
worries at all. If you have any more questions, just let me know. If you want me to read over something, just let me know as well. Like if you're
Cool.
not
Yeah. And if you're in Osaka or in Japan or whatever for some big neuroscience conference
oh
or
yeah,
whatever
let me know
to
if
do the
there's.
big release of your new drug or whatever. Yeah.
Let me know. I mean, I would love to go to Japan. Like again,
Yeah. Come on.
I think I should definitely. I'd make it. It's great to go to a conference. It's like one of the biggest excuses for you to like go to a conference and then go
Yeah.
vacation
Well,
at the same
Japan is like you mentioned. This is a great time to come because just the dollar is strong, the yen is weak. Inflation has not really hit Japan much either. I'm saying, yeah, we don't want to turn this into a weird little economy finance podcast. There's enough of those, and there are also enough of these days too. I'm realizing, but I'm jumping on that wagon because I do like this stuff and I like learning more about it, and connecting with people like you. So yeah, thanks so much. If people want to connect with you or they want to learn more about any of this stuff, where can they go?
time. You can email me.
Yeah. Oh, your email. Yeah, I love when people share their emails. It's like so heartwarming. Are there any like kind of cool little books or resources or YouTube channels or anything that you think, oh, this is a fun way to learn about the news of this or just the basic science?
I do read a lot of Nature Biotech. So I don't know,
Yeah.
do you have access to that? So sometimes Nature limits their
Nature, the journal, Nature
Nature
Biotech.
Biotech. Yeah, so it's like
Okay.
Nature is like the big, and then they have their
Yeah.
specific journals. Nature Biotech
Yeah.
is great because they released. I just sent you the world's first CRISPR therapy is approved. So they what they're really good at is that they'll release the research paper,
Uh-huh.
right, on their thing, and then they'll release an explanatory like size
Yeah.
community paper, and that's usually in their news. So there's like if you click Nature Biotech and you click their news, it's very great because they're like talking about like this one. There's like
Oh,
I don't know, like Nature. You click
yeah.
on the news, and they release news art. Like today, they released this one after obesity drugs success. Companies rush to skeletal muscle, stuff like this. So you click on it.
Hmm.
Sometimes it's for me. I get it for free because I am
Yeah, you're
I
part of
have
the
access
university
to
or
it.
something. Yeah.
But if you're willing to like,
Yeah,
sometimes
you pay a little bit. Yeah.
you're able to get them, and it's great because they'll talk about whatever's going on, and then they'll link the research paper, the actual primary research paper,
Yeah.
like microbiome made jet fuel. Like, okay, what is that? That looks cool. I'll click on it.
It's all linked
It's
up.
single,
Yeah.
yeah, yeah.
Cited, and because if you go to Google, normal things through Google or AI or whatever, you're just gotta trust it. You're like, okay, well, where are the studies exactly? That's
Like
that's
here,
that's perfect.
yeah. So this one's cool actually. Bioengineering, spider, silk. That's great. Like, oh, that's great.
Yeah.
So like it's very much like biotechy.
Yeah.
AI for drug discovery is booming, but who owns the something like that? So it's like you read these things, and then sometimes they'll link the raw research paper on it. Like
Yeah.
click drugs research prove their remarkable chemistry in humans. So you click on this and be like, oh, this is cool. Oh, here's the ALS one.
Okay.
Unprecedented blood biomarker enables ALS drug approval. Let's
Uh-huh.
see, I can send you that.
Yeah. Yeah,
So
yeah,
stuff
yeah.
like this, they
Okay.
usually would write,
This is like Christmas morning for me. I'm just getting all these
yeah.
thingies, these
It's like if you
articles.
read more, if you and you're like, oh, this looks cool, and of course
Yeah.
it's coming from a trusted, worthy source, right?
Yeah, Nature is one of the like three premier
Yeah.
journals or whatever.
And so then you just access it. You access the raw data, not the raw,
Well, I
the
don't know if I'm ready for that.
yeah. If you want to read the actual primary research paper,
Yeah.
I'll link it.
You want
And
to see the genome sequence and everything? No.
if you want. But yeah,
Yeah.
this is a good place to start to get. I mean, this one's a good one. Like I should probably read this right now. Like, but this was,
Yeah.
things like that. That's like very much like you
Uh-huh.
got the research paper on. They're writing this news release. It's called a news release. On it, and you read more about it, and then if you're interested, you can always like contact the first author of the
Hmm.
paper. The first author of the
Wow.
paper usually leads the investigation. So if I'm a first author, I lead it, and they're usually like
Okay.
postdocs or PhD students. So they're more than willing to like set up a call
And
and
there's ways to contact them.
yeah, on
It's
each
kind of
page,
there.
well, on each paper that you publish, you put out your email, your institute email.
Wow.
Yeah, yeah, yeah.
Dude, you just open up the whole new world for me.
Definitely. Like, yeah, that's where you have to look at the primary research paper, like not
Yeah.
the news release. The news release is written usually by the editor or like
Yeah,
somebody,
some
somebody
dude who's
on the
researching it
marketing,
for a couple days. Yeah.
or
Um,
they're trying to communicate your research for you. Scientists, we don't
yeah.
we don't write the news releases. They get
Yeah.
it for us. Yeah, usually.
Yeah, because it's a whole different way of communicating to the
Yeah,
public and
yeah,
SEO friendly, whatever it's in that. Yeah, yeah, yeah. You gave me your email. I don't want to put it into the show notes because then you're just gonna get eaten away by these alligators and sharks and things and weird spams and stuff. So can I link your I don't know a social media profile like LinkedIn or something onto the show notes or what should I use?
yeah. My LinkedIn's fine. I have a Twitter too.
Yeah. Okay. Yeah. Yeah, thank you so much for all this time. At first, I got the title of the call and it's like Michael, wait, 30 minutes. I'm like, okay, how am I gonna do this in 30 minutes? Let's rapid fire this thing. But luckily we didn't have to go there. We were able to even dig into some sort of personal stuff with Ramadan and with San Diego, Japan, and all that. So this is nice. Yeah, and now I got a little friend in San Diego.
Yeah,
Oh,
just
yeah,
stop by
great,
or kind of come or whatever. More than happy.
cool. Yeah.
Okay.
Okay, cool. All right. All right,
Uh,
thanks.
Sounds
See ya.
good. Take care. Bye
Bye.
bye.
Bye.